Okay, let's get started. And first, I want to make a few announcements. And as you see, we only have 2 lectures left for this semester. And we'll finish the translation of protein synthesis topic today, and by this week. And next week, so on our list schedule, it's on next Wednesday at 2:50. I will give a brief overview about the topics we would have for the final exam. And then next week, the TA, I talk with the TA, we'll have no recitations. So we'll be all done with the recitations by today. So you have one more homework and one more quiz. And don't forget to complete those homework and quiz. And also I want to make an announcement is that I believe starting from today, the course evaluation online will be open. And I saw a number of you attended the lectures throughout, and I encourage you to go there and provide your opinions about the class. And overall, I think this class, this is new approach, and we did everything online, but overall the class worked very well, and the grades are very good for most of the students. And I hope you can, everyone can work hard and do a good job for the final exam. And there will be, the final exam is scheduled on December the 4th, and also by eCampus at 11 to 1:30. It takes, it's 2-hour exam. I don't know exactly what's the time. I will look at, but it will be 2 hours. My idea is to have 50 questions, each for 3 points. And that is for, I'll see it is, yeah, I'll figure out what's the time. And so today we'll talk about protein synthesis. This is the last step of the information pathway. The information on the messenger RNA eventually will be translated into proteins, for example, enzymes or hemoglobins and antibodies. They will carry out various kind of functions coded by the gene in our genome. And so this is a schematic showing you the briefly what happens. You see, this is messenger RNA, and it should have a continuous information to guide the synthesis of a protein after splicing. And one of the very important player for the translation step is the ribosome, which is this green spheres. And you see, the ribosome actually it will bind to the messenger RNA. And then tRNA is carrying, you see, this is the tRNA. tRNA is carrying amino acid will come to bind to the ribosome. And then the amino acid will react with either another amino acid or with a growing polypeptide chain to form a protein. So this is the translation step. And you see this tRNA molecules. We'll talk about how tRNA molecules will be connected to the amino acids, and then how the ribosome catalyzes the formation of the peptide bond. And so here, actually, this is the structure of tRNA. They all look like very simple. They have this inverse L shape, 3-dimensional structure. And this here is a low resolution structure, electron microscopy structure of the ribosome with those tRNAs bound to it. And you see 3 of those have different colors bound to the interface of the large subunit and the small subunit of the ribosome. So in these 2 lectures, we'll talk about the details about how protein synthesis works in the cells, catalyzed by the ribosome. And so we'll start with the genetic codes, and we know the message on the spliced messenger RNA will be translated into protein. And this process need a tRNA as an adapter. So the tRNA, which have this anticodon codon recognition stem, it will actually recognizes the triplet codes on the messenger RNA. And then the amino acid connect to the tRNA. They will react with each other. Other peptide connect to tRNA. They will react with each other to form a peptide bond. And yeah, after the discovery of the double helix, then scientists immediately work on how to deciphering the genetic code. And I will not get to the details how it worked out, but it is mostly based on they are used some synthetic oligonucleotides. For example, poly-U code for phenylalanine, or another polynucleotide for code for another amino acids. For example, this I'll look at. I think they used one of those poly-C, which is code for proline. So if you add a poly-C and you add the cellular extract, and then you add amino acids, and then you see this in vitro system synthesize a polyproline. So it is based on those kind of approach. It is tricky, but the several scientists used various kind of synthetic oligonucleotides, and they deciphered all the genetic code. And so here are the 64 genetic code. And you see most of them code for amino acids. And 3 of them code for stop codons. And also, you see this graph is color-coded differently. The yellowish colored, those are the hydrophobic amino acids. And those blue-colored are the positive charged amino acids. The red-colored that are the negative amino acid, charged amino acids. And those purplish colored amino acids are hydrophilic amino acids. And you see, for all different kinds of species, all the proteins are formed by 20 different amino acids. And so this triplets of the genetic code, it can code for 64 different combinations. So some of the amino acids have like 1 code. And then a lot of others have more than 2 codes. For example, here I show you, like you can see leucine residue amino acid can have 6 genetic code for the same amino acids. And also, if you look at this carefully, for example, for these 4 different code for leucine, and the first 2 code are the same. The only difference between them are the last one. So that means even if, like, for example, if there's accident, code for the leucine residue changed to the last position, and then it will not cause a mutation of the amino acid, because these 4 code all code for the same amino acids. And for most of these, like, for example, if amino acids have multiple code, and you see the first 2 codes are the same, but the last 1 code is different. And so these are some of the properties of genetic code. And also one of the very important property of the genetic code is that what we call it standard genetic code, because all the different species of life, like from bacteria to mammalian human, we use the same set of genetic code to code for proteins. So this is the this same chart can be used to translate all forms of RNA into proteins. So we talked about RNA splicing in the last lecture. We know it is very important to have the intron spliced out accurately, and because you can see for the messenger RNA, you can have the spliced mature messenger RNA can have 3 possible reading frames. But in reality, when the protein gets translated, there's only 1 frame. It's 1 operating frame is translated. And if you read the sequence with the 2 other reading frame, and you encounter a stop codon like pretty quickly. So there's only 1 operating frame which actually code is which provide the code for the protein coded by that messenger RNA. So it is very important to not have a frame shift. So the triplet code will be 1 after another. Each of them code amino acids, and eventually by the end of that messenger RNA, there's a stop codon. Then this messenger RNA can be translated into a polypeptide chain and then carry out a certain function. So the reading frame is a very important concept. I hope everyone pay attention to this, especially if you're doing research or designing primers, doing molecular cloning, you are trying to express the fusion protein. You have to consider the reading frame. If you accidentally introduce one nucleotide in an open reading frame, and then it will cause a frame shift, and then it will cause like for example, a premature stop of the translation, or you can have a truncated protein which will not work. So we know tRNA are very important adapter molecule for between the messenger RNA and the amino acid. And so the tRNA, this is a schematic showing the structure of the tRNA. And you see it have this clover leaves kind of structure, and it have four stems. One is a D loop, and another one is T possessive loop, and another one is the anticodon loop. This is the loop which actually recognizes the code on the triplet code on the messenger RNA. And then all of them have this acceptor stem, which you see it have multiple nucleotides from a double-stranded RNA. And this stem is very important, actually, in the process for the tRNA coupling. So because this tRNA, it have to be coupled with the correct amino acid. Otherwise, the translation process cannot work properly. So the right tRNA molecule, for example, alanine tRNA have to be coupled to alanine before it can be used. And otherwise, it will cause problem in the translation process. And so this is schematic showing the structure of the tRNA. And this is actually the structure of, like if you look at the structure of the tRNA, most of them have this similar three-dimensional structure. This grayish colored region is the anticodon stem loop. And it has three nucleotides. Actually, it can form base pair with the messenger RNA. And then on this end, on the 3' end, it have a conserved, it have the CCA sequence, and which is actually, in eukaryotic, it's added by enzyme. In bacteria, it is like all the tRNA, when they got synthesized, they have this CCA sequence. And this amino acid is actually coupled to the 3' hydroxyl group of the last nucleotide. So we know we have there's 64 possible combinations of genetic code. Only 3 of them code for stop codons. All the others code for amino acids. But in our cells, we don't have that many tRNA to carry out the translation of the process, because some of the tRNA can recognize multiple codes on the messenger RNA. For example, for this, if a certain position, for example, the third position of a tRNA is a U, and then it can recognize a triplet code ending either with A or G. And if it ends with third code is C or A, it can only recognize G or U. But if there's a third code is a G, it can recognize 2. And if it is this like a unique nucleotide called unicycle, and then it can recognize 3 different kind of codes. So this actually allows the only limited number of tRNAs to translate all the genetic code for different proteins. For example, this anticodon GCI, it can recognize either CGA, CGU, or CGC, 3 of them. And so this kind of non-Watson Crick base pairing, it's not that strong, and it's not very stable. And so most of the codon and codon recognition count on the first two nucleotides, which form real Western Crick base pairing. And the third code does not form the conventional Western Crick base pairing, but this kind of recognition allows one tRNA to recognize multiple codes. And also this kind of interaction, it's not as strong as the real Western Crick base pairing, but it will allow the tRNA to bind onto certain strings so that the translational process can be happen in a very efficient way. So now let's look at some of details how amino acid get connected to tRNA. And so here I show you this is the general structure of amino acid tRNA. And you see this clover leaf kind of structure, that is schematic showing the tRNA. And the 3' end of this is the 3' end of the tRNA. And it have this CCA sequence. And this is the last nucleotide. This is the A of the CCA. And so the amino acid actually form ester bond with this 3' hydroxyl group of the last nucleotide, the A adenylate of this last nucleotide. And so this is very important process because only when the correct amino acid get connected to the correct tRNA, and then it will ensure the translation. In the translation step, a correct amino acid will be added to the growing polypeptide chain. So sometimes this is called the second genetic code because it is very critical in the make sure the translation of the messenger RNA in an accurate manner. And so two very important recognition steps happens in protein synthesis. The one step is the correct amino acid must be attached to the correct tRNA molecule by the enzyme called amino acid tRNA synthetase. And we'll talk about those enzymes in a moment. So this is the one step. So you add the correct amino acid to the correct tRNA. And this enzyme is involved in this process. This family of enzymes, actually. For every amino acid, there's an enzyme which recognize the tRNA and then add the correct amino acid to the 3' end of the tRNA molecule. And in the next step of the translation, the correctly coupled amino acid tRNA, it must be for base pairs, must be for pair with the correct codon on the messenger RNA and on the ribosome. And then the ribosome catalyzes the formation of peptide bond, and then translate the information on the messenger RNA into a polypeptide chain. So this is I'll give you a brief overview about the amino acid tRNA and how amino acid get connected, coupled to the amino acid tRNA, coupled to the tRNA molecule. And this is the two important steps for the correct translation of messenger RNA information into protein. And so, as you will see when we get into in the next lecture, the ribosome itself does not recognize the amino acid. It even does not recognize which tRNA is which. So, but the ribosome is capable of catalyzing the formation of polypeptide using the information on the messenger RNA. And the reason for that is because each of these tRNA is coupled to the correct amino acid by this enzyme, catalyzed by this enzyme, amino acid tRNA synthetase. So here, for different amino acids, there are two different families of enzymes, amino acid tRNA synthetase involved. And you see, for the class 1 amino acid, like adenine, cysteine, glutamic acid, they use class 1 amino acid, the amino acid tRNA synthetase. And so they have different kind of structure and the way they catalyze the reaction. But overall, they do the same job of coupling amino acid to a tRNA. And for this amino acid, like alanine, asparagine, aspartic acid, they use class 2. I'll show you the structure of these two different family of enzymes in a moment. And so here is what happens when the in the reaction between tRNA and the amino acid. And so in the first step, the amino acid actually react with adenylate triphosphate, ATP. You see, it is now coupled, the amino acid is coupled to the alpha phosphate of the adenylate. And then this amino acid adenylate, actually it will bind to the enzymes, amino acid tRNA synthetase. And so the enzyme will bind to a certain tRNA, and then it will catalyze the reaction. For example, for the class 1, it will first add the amino acid, transfer amino acid to the 2' hydroxy group of the adenylate. And then in the next step, the amino acid actually get transferred to the 3' hydroxy group of the tRNA. And this is the final product, which is amino acid tRNA molecule. And the region highlighted pink, that is the amino acid coupled to the C-terminus adenylate of the tRNA. And the class 2 enzymes actually do the reaction in a slightly different way. And it couples the amino acid directly to the 3' hydroxy group of the tRNA. And in one step, it forms this amino acid tRNA, which have the amino acid coupled to the 3' hydroxy group. But for the class 1 molecule, it is first the amino acid is added to the 2' hydroxy group, and then it is transferred to 3' hydroxy group. So that is the difference between the class 1 and the class 2 amino acid tRNA synthetase. Any questions before I move on? This is a very important part.
Dr. Lee, I do have a few questions.
Uh-huh.
So in the beginning, the amino acid is binding to the ATP, correct?
Yes, it is coupled to the alpha phosphate from this phosphodiester bond.
Okay, and then it's going to form that amino acyl AMP?
Yes.
Okay, so then that will go and bind it to the tRNA, correct?
No, then it will bind to the enzyme, and then the amino acid will be transferred to the tRNA.
Okay, so which is the enzyme? Oh, is it just the class 1 or the class 2?
Yes.
The blue. Okay, so it doesn't show the structure of it.
Yeah, this molecule and this molecule, they have to bind to that enzyme at the same time. And then the enzyme catalyzes the transfer of the amino acid, of this amino acid to the tRNA molecule.
Okay, that's where I was confused. And then so depending on if it's class 1 or class 2 will dictate which hydroxyl group it will bind to, correct?
First, yes, yeah.
Okay, thank you.
Other questions? So we know this is a very important process because the correct enzyme have to recognize the correct tRNA and then add the correct amino acid to that tRNA. So for each of the amino acid, and there's an amino acid tRNA synthetase. And that amino acid tRNA synthetase will recognize the tRNA, and then it will transfer the amino acid to the 3' hydroxy group of that tRNA. And so you see, this is the, I show you two structures. One is the class 1, and it is a glutamine tRNA synthetase from E. coli bacteria. You see, this is the tRNA bind to the enzyme, which is those green ribbons. And this is 3' end of the tRNA. That is the amino acid coupled to the adenylate. And then the enzyme catalyzes the reaction of transferring that amino acid to the 3' end of the tRNA molecule. And you see there's a pretty extensive interaction between the tRNA and the enzyme because this enzyme have to recognize the correct tRNA. And for example, this enzyme will not add this glutamine to a glutamate tRNA or alanine tRNA because it will not recognize that alanine tRNA. And it only have a surface which recognizes specifically this glutamine tRNA. And this is the second family of amino acid tRNA synthetase. And you see it's dimer. And also, similarly, it recognizes the tRNA, and then it transfers the amino acid from the adenylate, which is coupled to adenylate, to the 3' hydroxy group of the tRNA molecule. So these are the two families of enzymes which recognize the tRNA and then add the corrected amino acid to the 3' end of the tRNA molecule. And so because all the tRNA are very similar to each other, and actually a lot of nucleotides in the tRNA sequence are conserved. And so if you look at which kind of nucleotides got recognized by the enzyme, and so this is a statistical view, and you see it's mostly the acceptor stem. Those last few nucleotides on the acceptor stem, and then also at the 5' end of the tRNA, which play an important role in recognition. So you see, those nucleotides actually bind specifically to the enzyme. And also the anticodon region, which is here, which can also be recognized specifically by the enzyme. So that is how this enzyme actually added the correct amino acid to the tRNA, because they can recognize some of the unique sequence on the acceptor stem and on the anticodon stem, and then allow the amino acid to be coupled to the 3' end of the tRNA. And you see, if you look at this part of the molecule of the tRNA, it does not do anything with the enzyme, so they are not involved in the specific recognition. Actually, most of the nucleotide is not involved in the specific recognition. Only those big yellow sphere nucleotides, of course, and those yellow sphere region, they actually play an important role in recognizing by the amino acid tRNA synthetase. Is there any questions about this? So we know it's very important. The correct amino acid have to be coupled to the correct tRNA, and the enzyme actually recognize the tRNA and also recognize the amino acid coupled to the adenylate, and then it will catalyze the reaction of transferring that amino acid to this tRNA. And this process ensures every tRNA get coupled to the right amino acid. And actually, scientists are trying various kind of techniques, and they can actually introduce, for example, a different amino acid, which is not one of the 20. And then you can, in this process, you can actually add a certain unique amino acid, which is not nature, to the polygluing polypeptide chain. So this is possible. And so, but for the regular 20 amino acid, each of them have a certain tRNA, and then they have to be coupled to the correct amino acid before they can be used in the protein synthesis. So now I have to give you a brief overview. This is actually one of the very first few slides when we talk about protein. And so I'll give you a brief overview about how ribosome catalyze the reaction to form peptide bond. And you see, this is the gluing polypeptide. Let's assume we are looking at this process when in the middle of protein synthesis. And don't worry about how it is initiated. And we'll talk about how protein synthesis gets initiated in the next lecture. And now, let's see, this is the tRNA molecule, which bind to the P site of the ribosome. And it is coupled on the 3' end to a growing polypeptide chain. And then you see the next, on the next position, which is A site. And now you see that CCA coupled to a new amino acid. And then the ribosome actually brings this ester linked polypeptide very close to the amine group of that new amino acid coupled to tRNA and the A site. And then it catalyzes the reaction to allow the amine group to attack that ester bond. And then this polypeptide will be transferred to that amine group. And you see, once after this polypeptide got transferred to the ASAT tRNA. And then the ribosome will move by 3 code along the messenger RNA. And then this tRNA and the pSat will be released from the ribosome. And then the polypeptide chain, it will move to the pSat, which bound to the ASAT tRNA, will be translocated to the pSat. And then another tRNA coupled to amino acid will coming from base pair using the codon-anticodon recognition base pairing, and then another round of catalysis will happen. And until this ribosome will move along the messenger until it reaches stop codon. And then the polypeptide will be released from the ribosome. And then the ribosome will be recycled for another round of reaction. So now we know like ribosome is a very important player in protein synthesis. And it have the catalytic activity, which catalyze the formation of a peptide bond. So here I show you, these are the 2 electron microscope images of ribosome from bacteria and yeast. And overall, both of them contain a large subunit and small subunit. And in between, there's the RNA binding site. And also at the interface, the 3 tRNA bind to the ribosome. And so here are some of the details about the bacteria and the eukaryotic ribosomes. And so sometimes the bacterial ribosome is called 70S ribosome because this 70S corresponds to the sedimentation coefficient of the bacterial ribosome. And you see the eukaryotic ribosome is larger. It is called 80S ribosome. And the molecular weight of the 70S is about 3 million Dalton. But for the 80S, it's about 4 million Dalton. They are all very large molecules. And they contain a small subunit and a large subunit. Both of them contain a small subunit and large subunit. And I want you to pay attention to what's the component, other components of the ribosomes. And you see the large subunit, the 50S ribosome subunit, it contains 2 RNA molecules. One is 5S ribosome RNA, which have 120 nucleotides. And then the other one is the 23S ribosome RNA. It contains 3,200 nucleotides. So that's a very large RNA molecule, which actually form the scaffold of the ribosome. And also the large subunit contains 36 proteins. So it have a molecular weight of almost 2 million Dalton. And so the structure of the smaller subunit is simpler. It is the 30S subunit. It contains the 16S ribosome RNA. And this RNA contains 1,540 nucleotides. It's also a very large RNA molecule. And it also contains 21 proteins. So I'll not get to the details of the large subunit, but similarly, it contains multiple proteins, some very large, very important ribosome RNA molecule. For example, the 80S ribosome, 60S contains 4,700 nucleotide 28S ribosome RNA, which correspond to the 23S ribosome RNA. And so the small subunit contains 18S ribosome RNA, which is almost 2,000 residues, nucleotides, and which correspond to the 16S ribosome RNA. So,
Dr. Lee, I just have a quick question. Will we need to know those numbers, or do we just need to understand that the larger subunit
I think at least you need to know like what are the components of the large subunit and small subunit. And of course, in the next lecture, we'll talk about what are the functions of those.
Okay, so we'll know more about it next.
Yeah, at least you need to know, 23S ribosome RNA, that is the bacterial ribosome RNA of the large subunit.
Yes,
and then 16S ribosome RNA, that is the RNA of the small subunit of the bacterial ribosome.
Yes, sir. And will we need to
you
know
need to know the details about the eukaryotic ribosome, but at least you should know, it is the 80S. When we talk about 80S ribosome, that's eukaryotic, or for example, yeast or human ribosome.
Thank you.
Is there any other questions? Okay, so now let's have a look about the structure of the ribosome, because the structure of the ribosome is very important for us to understand how protein synthesis works in cells. And so this is actually, because I'm a structural biologist, and this the structure of the ribosome actually, I would say it is one of the highest achievement in the study of the crystallography field. And because these molecules are very large, very complicated, it is very, very challenging to solve its structures. But you see, as you see, and in 2009, these three scientists, Venki Ramakrishnan, Thomas Stats of Yale, and Ed Yonat of the Weizmann Institute of Israel. And these three scientists actually crystallize the ribosome subunits, and they solve the crystal structures, and which actually for the first time ever provided the molecular details about how ribosome looks like, and also provided insight how ribosome works. So they got Nobel Prize in 2009. And so here I show you some of a little section of the electron density map of the ribosome. And you see this is from the large subunit. And you see how those RNA fold and how they do form base pairs are very well resolved in this crystal structure. And all the protein, like you see the protein residues, they are pretty clear in this electron density map. So it is very important to get the molecular structure of the ribosome. Otherwise, we cannot understand how protein synthesis really works, but at molecular level. And so now I will move on to talk a little bit details about ribosome. And you see here, these are the two subunit, and this is the 50S ribosome subunit. That is the large subunit. And you see those grayish ribbons, those are the scaffold of the ribosome. These are the ribosome RNA molecules. And those colors like a sphere, like a globular protein, like proteins, they actually decorate the surface of the ribosome. And similar, you see 30S ribosome, and those grayish regions are the 16S ribosome RNA. And it folds into a very complicated three-dimensional structure and serves as a scaffold to allow many of those proteins to bind. And eventually it will combine with the large subunit and form a functional ribosome. So this is actually the 2000 picture from the Yale group about of the large subunit structure of the ribosome. And you see here, this is the 23S ribosome RNA, and it is a single RNA molecule, but it folds into this like 6-domain structure, and with different colors. And mostly it is through like the space pairing interactions, and they can fold into very complicated three-dimensional structure. And here you see this is a real structure, color-coded in a similar way, in the same way as this schematic showing how the ribosome RNA, 23S ribosome RNA fold. And you see there's a lot of regions, they form double helix by base pairing. And so a single RNA molecule, just as we saw with proteins, they can fold into a very large, very complicated three-dimensional structure. And to date, only RNA and polypeptide proteins can fold into unique three-dimensional structures. And actually the ribosome itself is the ribosome. It uses the RNA molecule to catalyze the formation of the peptide bond. So that is the large subunit of the bacterial ribosome. And so here I show you the structure of the small subunit. And this is the schematic showing you the secondary structure of the 16S ribosome RNA. And it is a lot simpler than large subunit because it's a way smaller RNA. But still, you see it can fold into 4 domains, each like have different color. And this is the actual structure, color-coded according to what we show here. And you see the red region, the 5' domain, it is the red region. And the green region, that is the central domain. And this yellow region is 3' major domain. And then this cyan color, that is the 3' minor domain. So you can see like RNA molecules can fold into very complicated 3-dimensional structures. Even though they only have 4 repeating units, and they can form base pairs by only 2 different ways, but they can fold into very complicated 3-dimensional structures. So here I'm showing the structure of the secondary structure of the 23S ribosome RNA again, and you see it forms multiple domains, mostly by base pairing in that single RNA molecule.
Dr. Lee,
Uh-huh.
I do have a quick question about the 23S RNA.
Uh-huh.
So that's what's interacting and allowing for peptide bonds to be formed, correct?
This is actually the large subunit contains the catalytic active site. You're right.
Okay, so okay. And what does the smaller subunit do? The
smaller subunit, like it is involved in the binding the RNA, and then it helps the codon anti-codon recognition, those kind of things. But the catalytic function of the ribosome is within this 23S ribosome RNA.
Yes, sir. And
we'll get there.
Also, okay.
Yeah. So here you see this. Here I show you. This is the 50S ribosome from this bacteria, and you see that is the ribosome RNA color coded according to we show in this picture, and you see that is how they fold into the complicated 3-dimensional structure. And so compared with the ribosome RNA, the proteins associated with ribosome actually looks quite small. And here I show you 2 different views of the protein which interact with a small subunit. This is the 30S ribosome RNA with those proteins in the like a rainbow color, and the ribosome RNA in the gray color. And similarly, you see like the 50S ribosome RNA, it also have a lot of proteins associated with on the surface of the large subunit. And so if we look at those proteins, they actually, most of them have unique structures. They have a globular domain, and they also have these long extensions, either a loop or a single polypeptide extension. This extensions play an important role in the interaction between the protein and the ribosome. And they tend to be conserved because they mediate specific interaction between the protein and the ribosome. So that is like, in the very beginning, those 3 scientists actually determined the structure of the 2 subunit unit. And with the information from electron microscopy, then scientists have pretty good idea about how the complete ribosome, the 70S ribosome looks like. But eventually, the 70S ribosome structure was also the complete ribosome structure. For example, from bacteria and from yeast were also determined. And also in the last couple of years, because of the power of the technology, development of electron microscopy, and many ribosomes from different species, either in the subunit or the full length, the 80S ribosome, they can be determined by at molecular detail. So now we know exactly how a complete ribosome looks like. And in some of the structures, they even have tRNAs in it. They even have the messenger RNA and 3 tRNAs bound to the ribosome. And here I show you how the tRNAs bound to the ribosome. This is actually a cut away view. And if you put these 2 together, that's a complete ribosome. If you open it up, and then these are the positions, the three tRNAs bound. And you see the decoding center is located on the 30S ribosome, which is small subunit. Actually, the 30S will bind messenger RNA, and then it will allow the tRNA to form base pair with that messenger RNA using the codon-anticodon interaction. And so the polypeptide, which is coupled to the tRNA, and the amino acid coupled to tRNA, they actually associate with the large subunit. So here I show you some of the details. This is maybe one of the very important image in this whole set, in this whole class. And I want you to pay attention to the 3 tRNAs bound to enter the A set, the P set, and the E set. So the E set is the tRNA, which is the amino acid is removed and is ready to leave. But the P set have a polypeptide bound to it, coupled to it. And you see by the 3' end of the P set tRNA, there's a big channel. That is like in the ribosome structure. And that channel is actually where the polypeptide come out from the ribosome. And also you see the tRNA at A set. And also its 3' end like positioned very close to the active site. It's positioned to the active site of ribosome, very close to the growing the ester bond between the tRNA and the P site tRNA. So the ribosome will catalyze the formation of a peptide bond between the amino acid on the A site tRNA and the polypeptide on the P site tRNA. So this is the active site of the ribosome. And so if we look at this structure very carefully, we not see any protein residues near the active site of the ribosome. That means the ribosome catalyzes the formation of the peptide bond by its ribosome RNA, by the large subunit, the 23S ribosome RNA molecule. So that is the structure of the active site of the ribosome. Is there any questions about this? So this is the 3' end of the tRNA at the P set and the A set. They are very close to each other. So the large subunit brings them very close to each other so that they can react with each other to form peptide bond. Once they form, when the peptide got formed, they will actually, the polypeptide chain, which is like extended, it will emerge from this channel. Eventually it will fold into a protein. So I will not get to the details. This is some of the structure details of the active site. And you see, this is what you see. And this is the A site, which you have amino acid coupled to the tRNA. And then the P site, which have peptide coupled to the tRNA. And then the ribosome actually catalyze the reaction between this amino acid amine group to allow it to launch a nucleophilic attack of the ester bond between the P site RNA, and eventually resulting in the transfer of that polypeptide to the tRNA and to the A site. And so this is the zoom-in view about the active site. So in this picture, I want to show you that black box, and here, which is exactly like what you see in that black box. And this is zoom-in, even a closer zoom-in, and you see that is the polypeptide coupled to the 3' of a tRNA. And then this is the amino acid tRNA, which have the amino acid. This is the N-terminal of that amino acid. And you see it is positioned right next to the ester bond between the polypeptide and the P site tRNA. And then this amine group will launch nucleophilic attack of that ester bond and eventually result in the formation of a peptide bond between the growing polypeptide and this amino acid. And so that is the catalytic mechanism of the ribosome. So in the next lecture, we'll get to a lot of more details about how protein synthesis works. And if you not know the details, don't worry, and we'll get to the points about to see how each tRNA gets delivered to the ribosome, and how the ribosome recognizes tRNA, and how it catalyzes the formation of peptide bond. That's pretty much what we have for today. And Is there any questions? We still have a few minutes.
Dr. Lee,
Uh-huh.
I have a question. What exactly is the reading frame?
Oh, you mean in the very beginning?
Yes, in the very beginning. I meant to ask, but I didn't want to interrupt.
So, for example, this is a section of RNA from messenger RNA. You can read the triplet like UAC, UAC, UAC like that. Or you can read another way, which is ACU, ACU, ACU. So,
oh, okay. So like for if
they
you were
code
doing
for different sequence of amino acids.
Oh, okay. And
But
so the first one,
in reality, for messenger RNA, you only have one reading frame. Because you only start with a certain HEG, AUG, and then you read triplets one by one, and then you complete the whole sequence of a protein. If you you reading a wrong reading frame, almost always the case after you read a couple of amino acids codes, and then you reach a stop code now.
Yes, sir. So you would start with the first reading frame and you just follow the first.
There's only one reading frame which starts from AUG.
Okay, so you wouldn't follow the ACU.
Yes.
Oh, okay, in the last lecture. Okay, I was just making sure.
You need to keep in mind if you are doing molecular cloning to do some lab work, you have to consider reading frame. Especially if you are fusing two segments together, you want to make sure they are in frame. If they are not in frame, you will reach a stop codon somewhere. And then your experiment will not work.
Yes, sir. So I guess like once there's a stop codon, that's when you would you would know when to start the first frame, correct? The first
frame always they start with AUG for the actual translation start from that AUG.
Yes, sir. And then you would follow from there the first reading frame.
Then you do the code AUG, for example, after that, UAC, UAC, UAC.
Oh, okay.
Yeah, that's really, it's a very important concept in molecular biology. And if you do not know that, you can easily make mistakes and your experiment will fail. Okay, that's a good question. Is there any other questions? So if there's no question, we'll stop there. And I encourage everyone of you, especially for those of you who like this class, and tell me why do you like it, and also like you if you have any suggestions so that we can do a better job for the future classes. I always welcome your suggestions. So do take some time, go to the course evaluation and provide your input so that the next year student will have a better experience with biochemistry 410. Okay, I'll stop there.